Good afternoon, everyone, and welcome to the SDB Ethel Browne Harvey Postdoctoral Seminar Series. It is my pleasure to introduce today's speaker, Dr. Martha Echevarría-Andino from University of Wisconsin. Today, she will be presenting her work entitled as HOX11 primes osteogenic enhancers during osteochondral differentiation. Dr. Martha began her scientific journey with a bachelor degree in biology from Puerto Rico University. Following her undergraduate training, she moved to University of Michigan, where she worked as a postdoc, and then she continued to pursue her PhD with Dr. Benjamin Allen in cell and developmental biology, where she looked for the function of hedgehog co-receptor during vertebrate embryogenesis. Later in 2021, she joined University of Wisconsin-Madison as a postdoctoral fellow in the lab of Dr. Denin Velic. Here, she is working on Hox transcription factors and their role in regulating extracellular matrix genes during osteochondrial differentiation. So today we will hear about her exciting postdoctoral research and the insight it provides into skeletal development. Please join me in welcoming Dr. Martha Echevarría-Andino.
Thanks, Gopal, for the nice introduction. As Gopal said, I would like to thank the organizers for giving me the opportunity to present my work today. And as Gopal said, I am currently a postdoc in the Willis lab at the University of Wisconsin-Madison. And in the Willick lab, we're interested in understanding the role of the Hox genes during development, repair, and regeneration using the musculoskeletal system as a model. And as many of you know, the Hox genes were initially discovered in Drosophila, and these genes are highly evolutionarily conserved, and they encode for transcription factors that regulate the regional patterning of the axial and appendicular skeleton. And specifically, I am interested in the HOX11 paralogus group, which patterns the sacral region of the skeleton and also the pseudopod region of the limb, which is my tissue of interest. The pseudopod is comprised by the radius and ulna in the forelimb and the tibia and fibula in the hind limb. And previous studies performed by Denin during her postdoc demonstrated that the deletion of the HOX11 paralogus genes result in severe skeletal malformations, as you can observe here in these skeletal preparations. To further understand HOX11 function during limb patterning and bone formation, previous studies from the lab were focused in trying to characterize HOX11 expressing cells utilizing a HOXA11 EGFP allele. Taking advantage of this allele, we were able to examine HOX expression throughout different stages. And here I'm showing you a longitudinal section of an E14.54 limb. And we were able to show that HOX expression is restricted to the pseudopod, specifically in the surrounding stroma and in the perichondrium of both radius and ulna. Surprisingly, the expression of HOX11 is not only restricted to developmental stages, and we can also detect HOX expression in postnatal and adult stages, also in the periosteum, endosteum, and in some cells in the bone marrow. Interestingly, the HOX expression cannot be detected in any differentiated cell types such as osteoblasts, osteoclasts, adipocytes, the vasculature, or the neurons, despite the striking phenotype that I showed you in the slide before. These observations led the lab to pursue whether HOX11 expressing cells were a population of mesenchymal progenitors. And to address this question, the lab pursued a series of rigorous studies over the last few years in which they demonstrated that HOX11 expressing cells are a population of cells that express progenitor-enriched mesenchymal stem cell surface markers such as PDGFR alpha, CD51, and leptin receptor. Additionally, we generated a lineage tracing system that in combination with the TD tomato reporter allowed us to follow these cells from embryonic stages all the way to all the way through the adulthood up to a year. And with this system, we were able to show that these cells are able to self-renew and also give rise to chondrocytes, osteocytes, and adipocytes. To examine the function of HOX11 in these cells, we also generated a conditional allele that allows us to perform conditional deletion at adult stages. And in these mutants, what we see is that um, HOX11 mutants display cortical bone defects, and they also display defects in the matrix, which contains highly disorganized collagen fibers. So to actually understand these defects in bone formation, we wanted to pursue if the HOX11 mutant skeletal stem cells display differentiation defects into the osteolineage. And to pursue this question, I isolated HOX11 expressing control mutant cells from adult and embryonic stages, and then we expanded them in vitro and subjected them to in vitro osteogenic differentiation. At the end of this essay, we stained with alizarin red to visualize calcium depositions, and as you can observe in here, both of our controls can actually form bone in vitro, but our mutant cells cannot. To further understand in which stage of differentiation these cells are getting stuck, we also wanted to examine the expression of different osteogenic differentiation markers. For this, we took advantage of some genetic tools that we have available in the lab that allow us to perform the conditional deletion and also lineage trace these cells at adult stages. We started by looking at the expression of RUNX2, which is an early osteoprogenitor marker, here in pink. And as you can observe, both control and mutants are able to express RUNX2. However, when we look at the expression of a late differentiation marker, such as osteocalcin, here shown in green, we see that only controls can express osteocalcin. And what this is suggesting is that our HOX11 mutant skeletal stem progenitor cells can actually exit the stem progenitor pool, start differentiation, and express early differentiation markers. However, they cannot fully mature and express late differentiation markers. So what my project is trying to understand is how HOX11 genes regulate skeletal stem progenitor cell differentiation into the osteochondral lineage. To address this question, we really wanted to know which genes are regulated by HOX11 that are required for proper osteogenic differentiation. And for this, we're taking a multiomic approach in which we want to know where HOX is binding in these cells. We want to know if HOX is regulating the chromatin landscape in these cells. And finally, which genes are differentially regulated by HOX function. I want to mention that this project is really big, and I've been working in optimizing different techniques and generating different large data sets. And right now, what I'm going to show you is work in progress, but I'm really excited to show you some new pieces of unpublished data. For time's sake, today I'm only going to focus on the HOX binding in skeletal stem cells and in the chromatin profiling experiments. But before we get to the data, I want to actually talk about the HOX transcriptional functions. So all HOX transcription factors contain a 60 amino acid DNA binding domain that binds to a 4-base core consensus motif that happens every 128 base pairs in the genome. And in mammals, there's a total of 39 mammalian HOX genes that have the same homeodomain and bind to the same sequence. So there's not a lot of specificity. And the specificity for elicit specific downstream targets is achieved through interactions with different transcription factors and cofactors such as PBS, PrEP, and mice. Additionally, there's not a lot known regarding the downstream targets for HOX genes. And one of the caveats, or one of the reasons that we don't know a lot about this, is because there is a lack of reliable antibodies to actually examine Hox binding. And additionally, in the context of skeletal stem cells, we don't know any targets or where it's binding. One of the only few known targets for HOX11 is 6-2, but it's in the kidney. So to circumvent these caveats, we generated two new alleles in which we gene edited a 3X flag tag in both exon 1 of HOXA11 and HOXD11. I want to mention that we focus in these two paralogs because are the ones that are expressed in the forelimb. So all the data that I will be showing today, it's in the forelimb. We validated these alleles by PCR and sequencing, and also we make sure that we could detect the expression of the flag tag by Western blot. And here you can observe that we can detect flag expression in both lines. And additionally, when we examine the expression by immunofluorescence, you can see that flag expression is detected in the surrounding stroma of both the radius and ulna in both lines. Additionally, we wanted to make sure that the addition of the 3X flag tag didn't disrupt the proper activity of the protein in vivo. So for this, we examined skeletal pattern, and specifically, since we wanted to look at the binding of both paralogs together, we generated double homozygous animals. And in here, you can observe that these animals display normal skeletal pattern, and when we quantify the radius and ulnar length, there's no significant differences in when compared to wild type. Next, we wanted to validate these alleles for DNA protein interaction analysis. So we took advantage of one of the few targets identified for HOX11, 6-2, and specifically in the kidney, HOX11 is expressed during embryonic stages, as you can observe here. We can also detect the expression with our flag alleles, and specifically it binds to an enhancer to control 6-2 expression. So taking advantage of this binding site, I isolated kidneys from E13.5 double homozygous flag animals, and I performed cotton run and cotton tag. And as you can observe here, I was able to detect enrichment using both techniques. So this was really exciting because it validated our tool and it allowed us to pursue the more interested experiments. So next, I utilized these tools to actually look at the binding of HOX11 in skeletal stem cells. And what we did is that we combine our flag alleles with our HOXA11 EGFP reporter. So we could isolate cells from postnatal day 0 animals and then sort them based on GFP expression and perform the cotton tag, which I optimized for this specific population. When we performed peak calling in our samples for flag using cotton tag, we saw that there were some variability among our biological replicates. So we decided that if a peak was present in at least 2 samples, this will be considered a true peak. And this led us to a total of 3,500 peaks. And here I'm showing you 3 different biological replicates. And as you can observe, we can get a very consistent signal across the 3 of them. Additionally, we perform mean motif analysis, and we get 3 main motif clusters, AP1, we get HOX, and then SP and KLF. Within the HOX cluster motif, we can find HOXA11 and HOXD11, and we can see that the binding motif is actually located in the central region of our peaks. Next, we wanted to annotate these peaks, and for this, we decided to use the genomic region enrichment annotation tool, which is a tool that actually links genomic regions to peaks, and also it will perform gene ontology analysis in data sets. Taking advantage of this tool, we were able to see that most of our peaks were at least associated to 2 genes per region. And additionally, we saw that most of our peaks are located in distal genomic regions relative to the transcriptional start site, suggesting that HOX could be, or it's binding to enhancer regions. When these peaks are annotated, we see that most of these regions are associated to genes that are related to cartilage development, connective tissue development, chondrocyte differentiation, extracellular structure organization, and extracellular matrix organization. And in here, I just want to show you a few examples of binding. So we can get peaks in COL1A1, SMOG-C2, SOLF2, DMP1, Indian hedgehog, GLE2, FGFR1, and BMPR1A. To further understand how HOGS was regulating these regions, we wanted to actually focus on analyzing the enhancer region since most of our peaks are there. So for this, we wanted to perform chromatin profiling in control and mutants skeletal stem cells. And for this, we use a similar approach to the one that we used before. So we isolated E18.5 control and mutant skeletal stem cells, and then we isolate them or we back sort them based on GFP expression. And then we performed cotton tag. So for this, we were focusing actually in histone modifications that will allow us to further refine those enhancer lists. So we were using H3K4 monomethylation to identify enhancer regions, H3K27 acetylation to identify active regions, and finally H3K27 trimethylation to identify repressed regions. So taking advantage of these data sets, we were able to show that about 78% of our peaks actually have the H3K4 monomethylation mark, indicating that they're enhancers. And if we combine the different histone modifications, we're able to further classify these enhancers into three different states. So an enhancer will be active if it has the H3K27 acetylation, so it's actively driving transcription. If it only has the enhancer mark, is an enhancer that it's prime and it's prepared for activation. And finally, if the enhancer has the enhancer mark and the H3K27 trimethylation, it's an enhancer that it's poised and it's currently repressed, but it can be activated later. When we look at the proportion of these different enhancer states in our control cells, we see that most of these enhancers are active, but then we have another good number of enhancers that are prime and a smaller number of enhancers that are poised. When we look in our mutant cells, what we saw is that mutant cells actually have decreased enhancer activity. And specifically, as you can observe in these heat maps, we can detect that they have lower levels of H3K4 monomethylation and H3K27 acetylation, meaning that they have less active and prime enhancers. When we look at the poise, we are not seeing any major differences, but we're also trying to incorporate ataxic data into this data set to further refine this. And there are many enhancers of interest, but we were mainly focused in enhancers that can actually regulate osteogenic differentiation. And to do this, I took advantage of some studies that recently have looked at the transcriptomic signature of osteoblasts and osteocytes. So I was able to generate a list of about 117 genes, and out of these 117 genes, HOGS bind to 50 of them. When I actually classify them through the different stages of osteogenic differentiation, we can see that HOX is binding to a large subset of these genes, but then most of these are actually genes associated with osteoblasts and osteocytes. And this is really exciting because if you remember from earlier in my talk, our HOX11 mutant skeletal stem cells are able to initiate differentiation, but they cannot fully mature. So we think that HOX is regulating the late enhancer program that is required for osteogenic differentiation. Here I'm showing you a few examples of some of these enhancers. I want to point out that all these genes are actually related to ECM organization, bone remodeling, osteocyte and dendritic processes, and some of these genes are also required for the transition from osteoblasts to osteocytes. And here I am showing you the flag binding, and then I'm showing you H3K4 monomethylation for control on mutants, and H3K27 acetylation for control on mutants. And all of these enhancers are prime because they do not have the active mark. These are just background levels. But what was really interesting is that the HOX11 control cells, we can detect H3K4 monomethylation and these enhancers, but we see that in mutants, the histone mark is significantly decreased. So what we think that it's happening in these sites is that actually HOX is binding um, to these late differentiation osteogenic enhancers to prime them and establish a regulatory landscape before differentiation. After these cells start differentiating, then another signal will activate these enhancers and complete the differentiation. And of course, in our mutants, we think that these enhancers are not being pre-established, so the regulatory landscape that is required for proper differentiation is not there. So, sorry, I'm having. So with that, today I show you that HOX11 primarily binds to these cell regulatory elements associated to cartilage and connective tissue development and extracellular matrix components. At the moment, we're trying to validate some of these targets, but we are really excited with the binding that we are seeing in these late osteogenic enhancers. Additionally, I show you that HOX11 mutant skeletal stem progenitor cells have decreased enhancer activity, and we think that HOX, again, it's priming all these enhancers that are going to be used during later differentiation. And finally, I didn't have time to show you this today, but we have some transcriptomic data that indicates that HOX function is required to regulate extracellular matrix components during osteogenic differentiation. So with that, I will take any questions. I would like to thank my PI, Dina Wellick, Paul Van Ginkel, a senior scientist in the lab. And I am very grateful for the mentoring of Jeff Billward and Marjorie Brand. And specifically, I want to point out Siri Yan, which is performing our bioinformatic analysis.
Great
So
talk.
thank you.
Yeah, great talk, Martha. Before we get questions in our Q&A, I would like to ask you a very kind of basic question. Maybe I have missed, but did you try to make double mutant where you just mutate both HOXD11 and HOXA11 and then look for the phenotype rather than doing separately?
So the ones that I have been showing, I show at the beginning, I double mutants for A11 and B11, and then the chromatin profiling studies that I'm doing in the mutants is in the double mutants as well.
Perfect, perfect. And by any chance, do you see some in your cut and run experiment, some reads coming in the non-coding region, which are kind of ultra-conjugate sequences, because some of them are known to work as enhancers?
Can you repeat the last part of the question? I didn't get it.
Yeah, so some of the non-coding sequences are known to work as enhancers. So did you see some peaks coming in those regions in your HOX mutants?
So yeah, we can detect enhancers in the mutants. We just think that the ones that are being bound by flag are not being primed, so they're not being like pre-established before differentiation. But yes, we do see enhancers in the mutants.
We have a question here. So it's from Timothy. So cool data. In addition to motif analysis, have you done footprinting to identify common motifs in very close proximity to HOXD11 motifs?
We haven't done that, but it's something that I can look into it.
Okay, thank you. So now another question. Do you see differences in binding profiles between the two paralogs?
So that's an interesting question. Unfortunately, I haven't looked at the binding profiles individually. So when we generated the alleles, the idea was that, we were going to look at the combined binding because they're so redundant and the phenotype only arises when we delete both of them. So maybe in the future I can look into that. But again, just based on the phenotype, we assume that it's binding to the same stuff.
Yeah, we do have another question. It's from Gage Crump. So, are the HOX11 dependent enhancer specific for the zygopod? Or if not, do other HOX or homeodomain proteins activate these enhancers in other part of the skeleton?
At the moment, we haven't compared the binding profiles with that we have generated with other ones. But I think that it's an interesting thing to do, and I definitely there's data out there, at least for HOX13, that it's available that I could use to do that.
Okay, let's see. So I don't see any other question coming. Okay, so okay, then let's thank Dr. Martha for her nice presentation. And now it's time for our next speaker, and let's listen her talk. Thank you, Martha.
Hi, everyone. My name is Neha. I'm very excited today to introduce Dr. Fernanda Palominos, who is a postdoctoral fellow at the University of California, Berkeley, where she works with Dr. Christopher Martin. Dr. Palominos studies the genetic and developmental mechanisms that drive craniofacial evolution using pupfish as a model system. She earned her PhD in Chile, where she investigated immune responses in zebrafish, the zebrafish olfactory system. Her work has been highly recognized, and she's won several awards, including NIH K99 award. I'm very much looking forward to her talk, and I hope you will join me in welcoming her. Thank you, Tanya.
Hey, thanks, Neha, for that great introduction. So I'll start sharing my screen. I hope that you can see the PowerPoint, and now that you will be able to see my presentation. So first of all, well, thanks, Neha, for that presentation. That was great. And I just want to say that I feel very humble and excited to have the opportunity to talk in this Ethel Browne Harvey seminar series. So this is the outline of my presentation. So I will give you a little introduction, and then we're gonna talk about like the work that I've been doing as a postdoc in the Martin Lab for the last 4 years and a half. First, we're going to talk about discovering novel craniofacial genes, and then how I've been validating their functions, and then, of course, conclusions. So, as an introduction, like the main question that I have in my research is trying to understand what are the genetic mechanisms behind extreme craniofacial evolution. And we can think about the craniofacial evolution from the perspective of comparing there you go, of comparing continuous craniofacial variation traits, such as the one that we are seeing here. So this is like continuous variation of the craniofacial features. But also we can include that and consider cases where like craniofacial traits can be extreme, which in many cases can lead, for example, to clinical variation that will impact someone's life and also performance. So here, for example, I'm showing you two skulls dating back to 1500 years ago. I'm showing you here a brachycephalic skull and a skull with prognatism. So this is showing you that there's historical clinical cases of craniofacial malformations in humans that we don't know a lot about. Although we can also see these extremes in craniofacial evolution or extreme in craniofacial abnormalities, as we put it in that way, in other vertebrates, such as, for example, dogs and cats. And you can see how different the craniofacial traits are. And this allow us, for example, to identify the different genes via genome-wide association mapping analysis. We have discovered several genes, for example, BMP3 and BMP6, and like having a function for giving these two different animals these different craniofacial traits. But even though we can understand the genetics behind and the evolutionary basis behind this craniofacial evolution using these mammalian models, we cannot understand the process of development, which is fundamental to understand how genetic variation is channeled into observable traits, such as craniofacial extremes, for example, as the ones that I'm showing you here. So, what I'm going to show you now is like this model where I'm addressing this novel knowledge gap. I am using this adaptive radiation of pupfishes that allows me to understand what is like the continuous or the normal craniofacial variation with like extremes as the ones that I'm showing you here. So here I have a pupfish that is brachycephalic, and I have two species of pupfish that have long elongated jaws. So the pupfish that I work with are endemic from San Salvador Island in the Bahamas. So this is the tiny island in the Bahamas. And they are amenable for lab rearing, as you are seeing them here, having like all this like shoe boxes with a bunch of like different fries. And here we have the tanks where we house them and we can breed them. You are seeing here a video of like several pupfishes moving their tails, and they get actually their name as pupfishes because they move their tails as puppies. The other thing that we've been doing with pupfishes and that we've been like pretty successful, here you're seeing like an egg of a pupfish. It's pretty similar in size of a zebrafish egg. And here we are collecting sperm that have facilitated us, for example, doing in vitro fertilization. So we can have like a population of synchronically developing embryos at the same developmental stage. As I told you before, also they have more or less like the same size of a zebrafish embryo. They are transparent throughout like embryonic development, although they have different pigmentation cells, such as the leucophores that you're seeing here, or like um, white pigment cells. So how different are the craniofacial morphologies? So the San Salvador Island pupfish specialists have extreme changes in their craniofacial morphology that are associated with their special dietary behavior. So if you remember in the first slide that I showed you, there was like the brachycephalic skull that have a snail symbol. That means that this pupfish eat snails. And then whenever you see this other symbol here, like scale, like a fish predating another fish, that will mean that that is a scale eating. So these two extremes of craniofacial morphologies are accompanied by this special type of dietary behaviors. So I'm going to play this so you can see the video of this pupfish eating their different food sources. So here you're seeing, for example, the molluskivore pupfish, like taking the snail out of their shell instead of like crushing the snail as a whole. And you can see also in this, the right side of your screen, you can see the scale eater pupfish eating or predating the scales from an euthanized zebrafish. And you can see how different are their craniofacial traits. We can see that, for example, in the case of the molluscivore, they have a pre-maxillary extension that we believe that is a evolutionary novelty that probably helps them take the snail out of their shell. And in contrary, you can see that the scale eater pupfish has much more teeth. They have like longer dentaries and wider jaws overall. And they also have increased aggression in terms of behavior. And at the same time, they have like a bigger muscle mass in the craniofacial structure. So all of this, like this huge this huge craniofacial divergence that we observe in pupfishes happens despite them having low genetic differentiation. That means that basically we can compare the genomes of this different pupfish species that live in the same island of San Salvador, and we can compare their genomes and try to identify the genetic basis for like this different craniofacial morphologies. So basically going back to the same question that I was like showing you before, now I want to answer this. What are the genetic mechanisms behind extreme craniofacial evolution in pupfishes? So now we're going to dig into some data that has been already published. And first, we're going to talk about discovering novel craniofacial genes and how I've done it in the past few years. So the aim of this part of this like 2.1 part of the talk is link genetic variation with gene expression changes during craniofacial development. And for that, what I did is like of the first comparative tissue-specific transcriptomic among San Salvador Island pupfishes and also their outgroup species. So here I'm showing you the picture of San Salvador Island and the different sampling locations where we took our fish from. And here you're seeing like the 4 different species and their craniofacial traits that associated uh, with the SSI pupfishes. So here we have the generalists that have like a generalist type of face. We have a molluscivore that is brachycephalic, and we have 2 different scale leaders, one that has like a much more prolonged and elongated jaw than the other. But we also included these 2 different outgroup species, a central and ancestral generalist from Mexico that has like around 100,000 years of divergence with the San Salvador Island pupfishes, and another generalist from North Carolina that has something like around 30,000 years of divergence with San Salvador Island, who have speciated super fast in less than 10,000 years. So we did that and we compared this comparative tissue-specific transcriptomic at the end of the embryonic development. And we compared 2 different tissues. We compared the jaw and we compared the tail. We wanted to compare these 2 different tissues because the jaw is a fast evolving tissue in our pupfish adaptive radiation versus a slow evolving tissue like the tail. Both of them differ, as we know, in embryonic origin, the jaw coming mostly from the neural crest and the tail coming mostly from like somatic mesoderm. But most importantly, because they also differ in their ecological relevance, being the jaw especially important for their dietary behaviors, this specialized type of dietary behaviors that they have, and the tail has like a more normal or common role in locomotion. So this is just like how many transcriptome we ended up sequencing, how many different species and population across geographical sampling sites, and the total billion raw reads that we generated. We did a bunch of analysis, but what I want to guide you specifically to this DE-SIG2 to compare between each one of the species, like this is the Atlantic generalist or the North Carolina generalist, and we compare here like the Atlantic generalist versus every other pupfish for the same type of craniofacial or caudal tail tissue. So here we have a volcano plot, and there's like a lot of genes that are up and down regulated, but we're not going to talk about that because I want you to focus specifically on this. So basically, if we pull together all the craniofacial differential gene expression that we found and all the caudal or log or tail gene differential gene expression, we can put that together and like have a specific percentage of differentially expressed genes that can be either craniofacial exclusive or caudal exclusive. So as you are seeing here, for example, in the North Carolina or Atlantic generalist, we are seeing like a very even percentage between what is differentially expressed and craniofacial exclusive, and between what is caudal or tail expressed and exclusively expressed, differentially expressed there. If we compare that with the inland generalist, that means like the generalist from San Salvador Island, we can see that the percentage, although we have a way less differential gene expression, you can see that there's like a even distribution, more or less, of the amount of craniofacial or caudal exclusive gene expression. But that is completely changed when we introduce or we look, for example, at the specialist. So here we're seeing the molluscivore and the scale leader, and you can see that around 90% of the differential gene expression that we found it to be specifically in the craniofacial region. So that means that there's high proportions of differential gene expression that are uniquely found in the craniofacial tissue of the specialist, the dietary specialist. And then what we could do, because we're working with like 2,000 genes or more or less, we wanted to narrow down the amount of genes that we had. So for that, we integrated this comparative tissue-specific transcriptomics with speciation genomics. So here we have, for example, some alleles that are fixed in this specialist that were identified through genome-wide association mapping analysis. And when we did that, we were able to narrow down our list in the case of the molluscivore to only 4 genes. In the case of the scale leader number 2, we were able to isolate or narrow down the list to 2 genes. And in the case of the scale leader number 1, we were able to get like 8 candidate genes. So this integration of known fixed alleles or fixed single nucleotide polymorphisms with craniofacial gene expression revealed 12 novel candidate genes that now we're working with. And I published this paper. We published this paper a couple of months ago, I would say like 6 or 5 months ago. And we were able to identify that, for example, this gene PISER3 that we found to be differentially expressed in the scale leader 1 and the molluscivore, we found that actually there's no differential expression, spatial differential expression between these two species when we look specifically at the oral apparatus. But if we look at the pharyngeal apparatus, we will see that, for example, this gene, PISER3, is highly expressed in the pharyngeal apparatus of the molluscivores, but not the scale leaders. So we found evidence of a spatial differential expression of PISER3 between our species at the end of embryonic development. And we also analyzed, for example, the expression of this other gene, ATP8A1. And we found that ATP8A1 also shows evidence of divergent gene expression at hatching. And here I'm showing you only the scale leader, but we can see that, for example, this orange gene, this orange label is ATP8A1 and is highly expressed in the intermandibular muscles of the scale leader. So a conclusion of this first part is that we have associated adaptive variants with craniofacial changes in gene expression, especially at the end of embryonic development. So that's work that is already published. But I also wanted to highlight the work of like one undergrad and the lab manager, Fifi and Malaya, that are working with this 12 candidate genes and to analyze their differential gene expression patterning spatially. So here you're seeing, for example, an immunofluorescence against collagen 2A1. So it's like the whole craniofacial skeleton or the ventral skeleton of the cranium. And here we can see another candidate that we know that has around 20 mutations in their enhancer region. And here we are seeing in the case of the scale leader that is like lighting the abductor muscles. So then for validating, we're going to talk about the validating their function. And what I did is like functional test, or what I want to do is functionally test the contribution of these novel genes during craniofacial development. So for that, I will talk about like another gene that we discovered that is important. This is like a previous paper from 2023, where you can see that in the case of the generalist, for this specific gene, GALR2, we have 2 SNPs that are fixed only in the scale leaders, but not in the generalist. So the idea of this is to understand how are these polymorphisms contributing to the divergent GALR2 expression during development, and therefore their craniofacial divergence. And we look at 2 times of development. We looked at the pharyngeal state and at the hatching stage. So at the pharyngeal, what we found is that GALR2 expression is decreased in the scale leaders at this stage. So here I'm showing you the generalist, the scale leaders, and the molluscivore. And you can see here that, for example, in both of these species, we have like a high expression of GALR2 in the most anterior part of the first pharyngeal arch, but we don't see that in the scale leaders. If we look, for example, at 2 different parts of the jaw at hatching, we could see that specifically we look, for example, here to the upper lip or the maxilla, we will see that at hatching, GALR2, now in orange, is found expressed in the chondrocytes of the megals and the palatoquadrate cartilage across the species. So that means that GALR2 is expressed in chondrocytes across all these like 3 different or 4 different species that we have in San Salvador Island. However, as I told you before, no, it's expressed in the megals and the palatoquadrate. But if we look specifically to the upper jaw, then we will see that the expression of GALR2 is only found in the premaxilla, as you can see here. So here we're seeing a bright field image of the premaxilla, where you can see GALR2 expression in the midline for both the scale leaders and molluscivores, but not generalists. So what we did to try to like identify what was the function of GALR2 during craniofacial development, what we did is that we used these two different pharmacological compounds, M35 and M871, that can target, for example, the two available galanin receptors that are in fish, GALR1 and GALR2, or a more specific inhibitor, which is M871. And we can see here that it's like inhibiting only GALR2. And what we did is that we basically incubated and allowed the fish to develop in either of these pharmacological agents, and then we assessed their specific phenotype and and other traits. So what we did with this pharmacological inhibition, and here I'm showing you a control and GALR2 a specific inhibitor, and you can see that the effect that we found was found exclusively in the specialist. And here I'm showing you one specific graph or one specific plot, the chondrocyte density. And you can see that basically the GALR1 and GALR1 plus GALR2 blocker or GALR2 blockers, the effect of chondrocyte density is only found in scalars, which means that GALR2 pharmacological inhibition, it's depending on the genetic background that we have, if it's either, for example, a generalist or scalators, because we don't see, for example, any change in their generalist or any change of on the molluscivore. And remember that the scalator is the only species that have this fixed SNP in the enhancer region of GALR2. So as conclusion of the second part is that we have validated the craniofacial function of GALR2 during embryonic development using pharmacological inhibition. And that work was also in collaboration with a fantastic lab manager that we had at the time, Vanessa Moole. But of course, like what we wanted to do is being able to perform CRISPR. So Heriberto has been working with me. He started as a summer undergraduate researcher last summer in the lab, and Heriberto has been helping me to create for the first time this CRISPR OCA2 mutant light in pupfishes. So here are you seeing like sequences of this OCA2 mutant where we can observe, for example, the lack of maturation of pigmentation cells in the mutant. And we found like a 4 base pair deletion in heterozygosity in this line. So that have been great, and we're looking forward to start CRISPRing our craniofacial genes. So as a conclusion of everything that I've been like walk you through, I hope that I have convinced you that I have a unique system to discover novel, fast evolving genetic changes that are associated with the development of extreme craniofacial variation that has evolved in less than 10,000 years. And that I can understand, for example, what is normal or continuous versus what is extreme. And there's so much potential to study and also study other aspects of their divergent craniofacial development, but also to compare with other different adaptive radiation of pupfishes. So this is myself looking for pupfishes in the Yucatan Peninsula like a year and a half ago. So that's also part of my research, bringing in novel fish systems to understand their craniofacial differences and their genetic basis of that. But I also collaborate with people in South America to understand, for example, their dramatic craniofacial differences in Orestias pupfishes from the altiplano, which are extremophiles. And this is like a personal project from Persilia, cryptic species in Chile that also differ in coloration and in craniofacial morphologies. So, of course, none of that will be able to be done without the amazing help of my PI, Chris Martin, and the people in the Martin Fish Speciation Lab. So, thank you. And I think I'll stop sharing.
Well, thank you for that really lovely talk. It was really, really beautiful. So to all the attendees, please put your questions in the question and answer box. And while we wait for that to get started, I have actually a couple of questions.
Okay.
So first, in your screen where you identified those 12 candidate genes, have you looked earlier in embryonic development to see if like maybe the timing of when they start to be expressed is different between your different species?
Yeah, we don't know anything. So right now, the work of Fifi and Malaya is basically taking these 12 candidate genes and like doing an ontogenetic series. Right now, we are just like trying with the hatching stage because it's like the easier, because mm, like zebrafish has like one chorion, like pupfishes have double chorion, so they are very thick. So the chorionating them is like a challenge for us. We've done some like pronase treatments to be able to decoordinate them more easily. But we're starting with a hatchling stage. But the idea is like go pretty early in development to understand temporal differences of expression of these 12 candidate genes that we know that they have mutations in their Hansen region, and at the same time they are differentially expressed. So I think it's like a pretty strong evidence that these genes are doing something that is important for their craniofacial development.
That's really awesome. I look forward to seeing that work.
Thank you.
The second question I have is just a little bit of a curiosity, and I was just wondering if there is anything known about galar in zebrafish.
Yeah, there are stuff. So galanin, it's a peptide secreted by the hypothalamus and is thought to mediate appetite control in fish and goldfish too, and to mediate somehow appetite control and aggression. So it makes sense that because the scale eaters are much more aggressive, um, that something is happening with Galar too. So we know that, for example, GALR2 is expressed in the chondrocytes, but we also know that it's expressed in the brain. So we, from another point of view, we're trying to assess also behaviorally, like what GALR2 can be doing for, concede more aggression in the scalar puffish.
Awesome. Okay, so we have another question. Can the scale eaters and the muscivores mate and produce live progeny?
They can, and actually it's super important. Like hybridization is super important for them to speciate. So there are other people in the lab that have worked with hybrids, and we have QTL mapping analysis up to the F2. So hybrids, they are actually they are not infertile, they are fertile. So we have, I think, up to like F3 hybrids from like different crosses, like generalis by scale eaters, generalis by molluscivores, and both specialists together. So we're now also trying to do a hybrid misexpression and allele specific expression too. So there's a lot of data that hopefully will come out in a couple of years.
And so our next question is, did you find any differences in the sensory systems between generalists, molluscivores, and scale eaters?
Oh, I love this question. Thank you. Thank you so much for that. Yeah, so there is sensory differences. We know that they're olfactory because my background is in olfactory development. That was one of the first things that I did when I came here. So we know that they do have like morphological differences in their olfactory rosettes. We know also that in terms of like brain lobules, they also differ in size. And in terms of neuromass, apparently they also are different. So neuromass or mechanosensory part of the lateral line of fishes, they do differ in patterning between this like 4 different species. Why is that the case? We don't know, but we look forward to look in more detail.
Questions? Well, if there's no other questions, I really want to just thank you both, Neha and Marta, for your excellent talks. The seminar was recorded and will be available on the SDB website next week. Please join us for next month's seminar on Friday, April 10th, when Joaquin Haciendo and Yixing Wang will present. Thank you all for coming, and goodbye.